Okay, we're going to go on to the sliding filament model of muscle contraction there on page 299 of your book. So you'll see this diagram there on page 300. What we've got at the top of the diagram here is, of course, a myosin molecule, a thick filament there in pink, and a single actin filament, myofilament right above it. Note that the tropomyosin troponin complexes are shown here. We've got several troponins, these 3 protein subunits that complex with this slender rod-shaped protein called tropomyosin. Note that the position of the tropomyosin troponin complex is blocking the myosin binding sites on the actin. You can barely see those little pink structures there. If you look super close, they're kind of hiding, but they're being blocked by the blue colored tropomyosin. So there's no opportunity for the actin and the myosin to interact with one another. However, when we introduce calcium ions into the equation, if you will, and you might remember, where are those being stored? In the muscle fiber. We talked about that in one of the earlier videos. Remember, it's the sarcoplasmic reticula, those extensive networking of little tubes in between all the myofibrils, where the cell stores the calcium. And in response to a muscle impulse that's generated at the neuromuscular junction that races down the sarcolemma, goes into those T-tubules we talked about. Remember the cisternae on either side of the T-tubule? That results, when that muscle impulse comes into the T-tubules, it causes the sarcoplasmic reticula to release its calcium, and that calcium goes out into the myofibrils. That's where we are now. We're at the sarcomeres, right? And so when that calcium is released, the first thing that it does here is it bonds to the troponin. Remember the 3 subunit protein that we see scattered here and there. So there's the calcium in sort of light yellow. It bonds to the troponin, and it causes the tropomyosin to slide off to the side. And of course, what do you see has happened with respect to the myosin binding sites on the actin? Well, they're now exposed, right? They're open. They're available to interact with those cross bridges of the myosin. And that's indeed what's going to happen next. So the calcium binds to the troponin, the tropomyosin moves off to the side, the myosin binding sites on the actin are exposed. Let's just kind of work around our little cycle here. Okay, the first thing that happens next is these cross bridges, note they have ADP and phosphate listed. This forms as a result of an ATP that was recently broken apart. You might remember from earlier parts of the lecture we talked about how when ATP is broken down, it forms ADP and phosphate. So that's where that comes from. And it's on the surface of these little kind of projections, these little cross bridges that are sticking out in all directions from the myosin filament. And so now we have the cross bridges coming up and contacting some of those binding sites on the actin. Well, after that happens, the ADP and phosphate will leave. They disassociate with the cross bridges, and the cross bridge undergoes what's called a power stroke. Notice it's going to pull the actin, in this case, to the left, because we're looking at the right-hand side of the sarcomere. If we were on the left side of the sarcomere, that actin would get pulled to the right. So this is pulling the actin to the left. More ATP is brought aboard thanks to the abundant mitochondria. And when that ATP comes on board and touches these cross bridges, that results in them breaking off of the actin. They disassociate, so the link is released. And then notice what happens to the ATP. it's broken down back into ADP and phosphate, and that causes these cross bridges to recock. And then what are they going to do? Well, very likely go back up, grab on to another part of the actin. The ADP and phosphate leave. We have another power stroke. We pull the actin, in this case, to the left even more. We bring on more ATP and break it down. We disassociate the myosin from the actin. The ATP is degraded. The cross bridge recocks, and we keep doing that again and again and again, as long as we have plenty of energy, until the sarcomere goes from a relaxed state to a contracted state. So I want you to watch this video that I've posted. You won't be able to watch it on the voiceover PowerPoint, but if you go back into the regular PowerPoint for Chapter 9, that video will play. And then, of course, I also have some Khan Academy videos. You've seen a few of those perhaps. Here's another link to another Khan Academy video if you want to watch that. That talks about this very interesting process that we refer to as the sliding filament model that explains skeletal muscle contraction. This next slide is showing, of course, at the top here, a relaxed sarcomere, right? There's the myosin in pink. Here's the blue actin. And we talked about the interaction of the cross bridge, remember, of the myosin with the actin, how it goes and grabs on. And these actin are going to get pulled to the left. And these guys are going to get pulled to the right. The overall effect is to do what to the Z lines? They're getting closer together, aren't they? Yeah. Now, here's a question for you. Does the myosin or the actin filaments themselves change in overall length? Yes or no? The answer is no. If you look at this myosin, is it any different in length than during the relaxed state? No, it's the same length. And likewise, is the actin down here on the right side, is it any shorter or longer than in the relaxed state? Answer is no. It's the interaction of the two, right? As the myosin pulls the actin, and in this case to the left, or in this case to the right, that the overall effect is to shorten the sarcomere. And that's what we're seeing here in the lower part of the diagram. This is just a photograph from a stained skeletal muscle section. And you can see the Z lines here on the sides. And again, what happens during contraction is the Z lines get closer together because we're interacting that myosin and the actin. All you got to do is make sure you got plenty of calcium and you got plenty of ATP, and you can contract the muscle fiber. You can contract all of these sarcomeres. Remember, there's tons of sarcomeres in each myofibril, and so when each one contracts, the overall effect is to shorten the myofibril, and if you shorten all the myofibrils, you shorten the muscle fiber. What about relaxation? What happens during that process? Well, you've got to think back to where this whole thing started. Where was that? That was at the neuromuscular junction, right? So if we could stop the release of the acetylcholine at the cleft, we would not generate a muscle impulse. If we don't generate a muscle impulse, we won't have contraction, and the sliding filament process won't take place. So to stop a muscle from contracting, to let it relax, you've got to go back and junction. Okay, and that's what this right-hand part of this table is talking about. This is the steps involved in relaxation. Here we've got the steps involved in contraction, which we just talked about. So back in the neuromuscular junction, there's an enzyme here that's released called acetylcholinesterase. I know it's an enzyme because it ends in ASE. Remember, all enzymes, or any, I should say all words that end in ASE are enzymes. Not all enzymes end in ASE, but if you end in an ASE, you are definitely an enzyme. So this particular enzyme is released at that neuromuscular junction, as the video talked about, and that breaks down the acetylcholine if you break down the acetylcholine, it won't have any way to combine with the receptor there on the folded sarcolemma, so you won't generate a muscle impulse. If you don't generate a muscle impulse, then the calcium ions that were released, remember, in response to a muscle impulse, they're no longer stimulated to be released by the SR. And so what does the SR do? Well, the sarcoplasmic reticulum, notice what it says here, it pumps that calcium back into that organelle. Active transport, remember that? Movement of materials from a region of low concentration to a region of high concentration, just the opposite of diffusion. That takes energy, doesn't it, to do that? So when you remove the calcium, when you pump it back into the SR, or sarcoplasmic reticulum, you then remove it from the troponin, and if the troponin doesn't have calcium connected to it, what's going to happen to the tropomyosin? It's going to move over the surface of those myosin binding sites, right, of the actin, and you're going to be blocking those myosin binding sites. So you can't have any interaction between the myosin and the actin. And if you can't have any interaction between the two, what's going to happen? Well, the muscle fiber is going to relax. So spend some time reviewing these two very important parts or events within contraction and relaxation. These are absolutely critical to know. Also in Blackboard, I have posted a super excellent animation. It's within the PowerPoint listing. I want you definitely all to go back and watch this. It takes you from the very beginning of the anatomical aspect of skeletal muscle structure all the way down through contraction and relaxation. It's really, really, really good. So please make sure you watch that.